I left the SIB 3 years ago now. I can actually mention some of the past work because I spent about 11 years there. And also try to echo some of the presentation that was happening this morning as well. When Abdullah actually asked me if I could give a talk, I was hoping by that time now, we would have a very nice story about multi-omics and integrative biology, but somehow a bloody nasty virus came around and a lot of our activity were refocused, like a lot of people worldwide and in Switzerland in particular, towards either following this virus genetically or actually following the vaccine and the vaccination part. So I'm currently having several jobs, but my first job is to be a professor at the University of Lausanne and at the University of Geneva because it's a cheap way to teach without hiring a new professor. I wish that they hire a new professor. So if anybody is recorded, that would actually go and push the University of Geneva to hire more professors, that I would be more than happy to help in that stuff. The activity, however, that I spent the last 3 years was really trying to help the Genome Center, which is located in Geneva. And I'll talk about this because it's part of the PHRT platform that has been created 4 years ago, and part of the ECHO sister project called SPHN that we heard part of it by Mark earlier today. So one thing I'm always joking when I talk to clinicians, I've been talking to clinicians both when I was in Saronno for about 8 years, from the key opinion leaders that we were paying very dearly to give us insight about what our next target value was, to the biologist when we were actually providing information. They all of them wants to have a red big enabling button. I showed that to George Kukos at the Ludwig, and he really laughed about this because fundamentally that's what we are all aiming at. Somehow you could put behind this red button AI or whatever buzzword people could get their job with nowadays. But at the end of the day, there's a lot of things that is happening behind, and it's kind of tantalizing to look at this picture. I've been using it for systems biology, been using it for bioinformatics, could be used for AI, but at the end of the day, the vast majority of the time, except that when we want to tell a story or write a paper, the answer is most of the time, no, it doesn't work. Now I'm going to show you a few where it works, and somehow I feel always that there is part of a luck and part of really that we understand how it works, but most of the time it's really luck that strikes. The first example is something that is a real world implementation. And I added that mainly because we haven't talked about things that already are successfully implemented that transform completely the way we do diagnosis. And this is an NIPT, a non-invasive prenatal test that we developed some years ago, have been put. And this is the only time, and I remember Daniel Stekoven sometimes talking about his wife. I'm also talking about my wife most of the time. This is the first time that my wife was really happy that she married a guy that was doing a bit of something useful. The other aspect I'd like to discuss, and this is the second topic, is modeling and simulation, where we're actually trying to use a drug that was inefficacious and try to repurpose that in triple negative breast cancer, not to target the cancer cells, because ultimately a lot of the cancer disease are an immune problems. And if you train the immune system, you kill the cancer. And that's the beauty of the IO intervention that got Nobel Prize and whatnot. The last part, and I'm going to talk about this in the last few minutes, is implementing clinical grade sequencing into Health 2030 Genome Center in Geneva and trying to assess whether we are ready to do this multi-omics that everybody talks about and pretty much the red button discussion. The non-invasive prenatal test basically started with 4 components. One was Professor Nikolaidis, that is also Greek like me, and has been working and developing the nuchal imaging for fetus that bears trisomy 21. Has been working for ages onto this, and molecular genetics or molecular diagnostic that would not had the problem of getting a needle inside the womb of the mom was something that he was looking for quite some time. 2 people, one was the one that is behind some of the early patents of Illumina. That's Laurent Farinelli at Fasteris in Geneva, and Bernard Corrado is a medical geneticist working also on trisomy 21 for quite some time during his first years, with also the other partners into this one. And the two people that are not anymore at the SIB, but part of the BICS, which is the Bioinformatics Competence Center in Lausanne, at the University of Lausanne, the EPFL, have been designing the algorithm. And at that time, we all were at the SIP, with the aim to create an algorithm that allows to do a pure detection with a validation and accreditation for this. As you all know, and you might know, there are several routes by which an invasive procedure was necessary to test if the mother had a risk of trisomy. And there are several measures. The imaging was one. It was actually painful. It's actually something that carry a risk of miscarriage. But basically, this is a very invasive one. The change, which pretty much dated back in 2008 when Stephen Quake and some of our postdocs actually developed a method for sequencing the blood of the mother and the baby, allowed to actually count which chromosome or how many chromosomes are in the blood. And this is actually a very well-known biological fundamentals that actually overtrained athletes, cancer patients, and Mark alluded to that during the discussions earlier today, and babies or fetus actually deliver blood and DNA into the blood as cell-free DNA. So being able to sequence that allows to detect it. So there were already existing technology, but one of the questions was, most of them were working very well for trisomy 21. What we tried to do is actually to reverse a bit the problem to have a detection rate that was higher. So it's a complete different algorithm compared to what has been published by Stephen and patented by Stephen Quake, by the way, mind you, which allows to detect trisomy 21, 18, 13, and 22. In this kind of cases, we always know the answer, so it's easy, right? We could treat the parameters so that it actually goes into the right direction. What it becomes more complicated is when you test it into a double-blind clinical trials, and this is where sample provided by Nicolaides in King's College, where basically we tried to test which type of trisomy and other aneuploidies we were detecting. And to be honest, I've been in this business for quite some time, and I didn't believe that we were reaching, and it's actually not thousands of samples, it's still a handful, but enough for clinical trials for this kind of technique. We were reaching 100% specificity and about 100% specificity except for one. And to cut a long story short, the one that we missed, which is the trisomy 18, with a low fetal fraction of DNA into the blood. The bottom line of this is also how fast this has changed the game of prenatal diagnostic. It changed the game because in 2012, in June 2012, the company MediSupport came to us. In October, we calibrated the new methods because we implemented what was published in Stephen Quake's paper. It works quite sort of okay for 21, but not for the others. And in December 2012, we had the validation for the double-blind experiments. We reached the CE marking in April 2013, and in May, it was on the Swiss market. The interesting part is that because it became a change in clinical practice for the gynecologists, and also it became kind of a norm for parents who are stressed about genetic defects, and to actually use that test, the LAMAL, which is the health insurance in Switzerland, has reimbursed that since 2014, sorry, if you have a high risk for this. Otherwise, it's actually something that you have to pay out of your pocket. So this is actually an example of a project that has turned into a real one. And I like Victor's presentation before because people are drowning in data. I think one thing that is quite clear is that you drown into the data and when you don't know what to do with it. The day you know what to do with it, you're going to change and transform it so that it's actually interpretable correctly. And honestly, with the flurry of the variant of unknown significance that we have, we are drawn mainly into the lack of our knowledge rather than anything else. And the data set is that we can't really interpret that well. Or we interpret the ones that could be only reimbursed, as Valerie showed that very nicely this morning. To the conclusion, the NIPT has been implemented worldwide, basically from China to Europe to the US. It's a simple blood drawn. It allows to assess the status of the embryo. And it generates roughly about $5 billion in revenue per year. So that's actually quite a good stuff because, hey, what? We reproduce as a human species. A fraction of cases are still complicated, and those ones I don't show because I'm only showing the stuff that works here. But there is a lot of cases where either the twins, the ethnicity, or the BMI plays a nasty role into this. Now, let me go to the second example of second vignette, if you wish, of trying to repurpose or transform the way we were doing this. So this is a project that started with Marie-Agnès Doucet that was in George Cukos' lab as an independent researcher and now is leading the oncology for ICNOS, which is a company up in Epalange. Nicolas, who is now at the BIX, and Isaac, who moved to the Ludwig Institute of Cancer Research with George Cukos. And people from the breast cancer center, Jean-François Delahaye, in the pathology, because you always need a pathologist, as we have heard, either from Mark or Victor, that we always need a pathologist. And they are actually an important player into that game, and actually quite one that brings us back to the ground, as we heard earlier with the presentation from Victor. So the whole idea behind this was to say, all right, can we try to use modeling as a way to tie things together rather than having listed things that goes up, a heat map of things that are green and red and of different colors? Can we actually try to model things and put that into context? Well, you can actually try to do AI or machine learning and try to do generative models, which most of the time are the acyclic graph, which actually is fine when you have a full differentiation process. But when you have homeostasis, which is the reason why we live, not differentiation process, is actually most of these methods fail in this case. So in Expect, what we have been trying to do is to gather the literature, build up models, which actually are rather small. And you don't need to be comprehensive for this. I mean, hopefully you have all the items that you want into the models and interrogate those models to do the what if. By no means we believe that these models are ground truth, and by no means we believe that they are representing reality. They are just toys to interrogate. So in this case, we were actually playing with type 2 expressing monocytes, which are actually increasing breast cancer patients and infiltrating in the tumor. And they actually infiltrate at a high rate. And one of the questions was to say, could we identify either what pushed them to become these infiltrating monocytes, or could we find a combination therapy that allows us to reduce some of their hallmark markers like type 2 VEGFR, for example? So in order to do this, the first phase was to phenotypically characterize those ones. We were doing that by fax, look at pro-angiogenic activity. So you could actually do that in vitro and in vivo as well in the eyes of mouse that do not have vessels. You could do a cytokine secretion profile for this, or a gene expression profile. This was the only molecular stuff which we used, which was mainly RNA-seq. We put that together into dynamical models and trying to identify what would be the best set of combination to move a set of receptors, in this case type 2 and VEGFR, and modulate those guys. So this is actually what we got from purely deriving the data that we have acquired on this by just treating with a set of 12 combinations. And you could see that there are molecules in there that are actually regulating either positively or negatively. There are a few feedback loops that are on them, one of them being the PLGF one. And there is a small dot, which is an end logical gate, which says that these two together needs to happen together. By using this model, which is, honestly, you can make a bit out of biochemistry, and I've been doing quite a bit of biochemistry, but most of them, I don't understand why this is data-driven from what we have been measuring. One of the questions, however, is to say, okay, I'd like to push these type 2 monocytes into these two classes. One is the peripheral type 2 monocyte, the ones that are not aggressive, and the ones that are tumor resident monocytes, which have as hallmark a high VEGFR1 and a high type 2. Now, the models was predicting 11 therapeutic interventions. So that's quite a lot, of which one was attracting our attention, which was a PLGF, TGF-beta, and type 2 inhibition. Good for us, we had type 2 inhibition that failed in clinics because of lack of efficacy. However, on the other side, and this is more a diagnostic prediction, the PLGF-NH2-TNF was actually the one that was predicting to move the peripheral blood 10 to the tumor resident one. So to cut a very long story short, and this is all published and you can find it in the PLOS computational biology paper, we actually tested our hypothesis. Now, obviously, when you have three combination therapies, you can do the checkerboard. This becomes a bit of a crazy experiment to do. So in this case, what we were mainly doing was trying to take the resting monocyte and actually transform them into the first, which we could call the progenic one, the PLGF-Ng2 TNF. And sure enough, the VEGFR and Tie2 expression were skyrocketing up. That's to the point that we had to place it at 100%. We had also an angiogenesis assay, which actually shows us that it was actually increasing. On the other side, we use actually our prediction, which is this little guy here. So angiogenetically speaking, you have a reduction, clearly, quite a lot of variation, but that's normal. And from an expression profile, we can actually see the reduction of this by using this combo therapy. The Tie2, on the other hand, lacked any efficacy. This we knew. And at that time, the patent for the Tie2 were off, and we actually filed a patent for this triple therapy for breast cancer, which is now actually published publicly. So the conclusion for this has been that we could identify through modeling a set of testable combination therapy. I think this is an example of what I think and I hope we'll move towards in the future. The lack of Tie2 inhibitors that was showing efficacy demonstrated effect, and we have done that ex tempo from the monocyte that were extracted from the breast cancer tumor through the Jean-François Delalay at the CHUV in vivo and in vitro. Now let me, in the last few minutes, go to the last part, which is trying to create the right environment in Switzerland. I mean, Switzerland, as we've seen from the COVID situation, and we hear and see NZZ, Le Temps, and others, is digitally incapable. However, we have all the means, money-wise, but we tend to create a lot of small castle left and right, because everybody buys their sequencer, everybody buys their digital pathology stuff. And at the end of the day, that's fine, because they need to care about the patients that comes into their class. But the day we wanted to do that at scale, and we want to digitize clinical samples with the latest technology, it's becoming not something that the University of Zurich can do, or the hospital at the CHUV where I'm located can do. So the idea of the PHRT was trying to create, and really I'm putting the trying because that's what we have been trying to create, is the next frontier for digitizing clinical sample with WGS, West to a certain extent, but I say we heard from Hugo that this is going to be dropped off soon, and I hope the sooner the better, because it's a weapon of mass distraction. RNA-seq, epigenetics, Mark talked about this earlier, on the genomic side, on the proteomic side, which is mainly in Zurich, quantitation and PTMs, and that was under the guidance first of Rudi Ebersold and then Bernd Wolscheid. And the metabolomics is mainly the Nicolas Zamboni. We are not at the clinical grade for the last two, because it cannot be entered in clinical practice. But as we've seen from this morning, and as I showed you from the NIPT, this is coming from the genomic side much more easier, because technology-wise, it's more robust. The need for clinical sample handling is the thing that matters the most. And the data generation has to be comprehensive and cost-effective, because at the end of the day, you don't want to pay like 5,000 Swiss francs for a BRCA test into a hospital when you can actually do that through a WGS of your germline and low coverage of your tumor. So the Health 2030 Genome Center that is located in Geneva is now seeking the ISO 15189 accreditation, which we should have, but I'm waiting for the fax from Bern because they seems to send things by fax for whole genome sequencing, whole exome, and RNA-seq. If you want to know more about this on the 29th, I will give a bit more detail on the interpretation analysis aspect, and we'll have a Swiss Genomic Forum in June this year, probably by Zoom, or perhaps if vaccination goes fast, which is doubtful, by in person. Now, the last part is when we have all these technologies, we want them to be brought together. In this case, what I claim here is that we lack a lot of methodologies, and it's not AI that's going to solve that, at least to my taste. It's mainly a way to attract funding lately. And if I look at the COVID case, all the 21 or 30 AI stuff were showing ridiculous predictions, and actually where none of them were used. The prostate is a good example, however. But one thing that is clear is that this looks very cool on the slide, but it's actually quite complicated to implement. And I would advocate that we don't have, at the time, the right methods that have been developed. And also the real challenge, intellectual challenge, to see whether these methods work effectively or not. Because we always publish papers, but at the end of the day, it's just one for our CV. The implementation is another story, as we heard today. So one of the implementation projects that we did for the KISP, the Kinderspital in Zurich, was from Professor Baumgartner, who has been using and dedicated all his entire life to the methylmalonic acidemia, which is a metabolic disease which disrupts amino acid metabolism. He collected over the years about 250 samples from around Europe. And one of the questions that was raised there was, can we have a disease stratification from the multi-omics? Can we explain and identify additional genes beside the MMA, which is the most known one, which is a rare disease in that case? And how much ground do we gain by mixing metabo, proteo, and genomics? To some of these questions we have answered, but some others, like the last one, I don't think we have a real clear answer at the current time. And I would say it's going to take quite a lot of time, even for this small data set, to be able to churn it and turn it and transform it. But to cut a long story short, and this is the work from several people, of which David Lampata was part of the House 2030, to switch with my stuff, sorry. House 2030 Genome Center is now at Roche, or Ximena Bonilla, who is in Gunnar's Resch lab, and Cheng Wang Guo, who was actually in Patrick Pedrioli and Bernd Wolscheid, almost said Bernd Weidenmiller, places at the CPAC, and Patrick Fournier and Sean Crozy. So all this was really trying to look at the mutation, and sure enough, we found more mutation than you would get in normal or even targeted sequencing. We characterize about 5,000 proteins, and that's actually quite a lot if you think from a proteomics point of view. Identified also a few QTL-based analysis, which actually are genetic variants that enhance or reduce the effects of a transcript or a protein. All that to say that this was done and generated very fast, because the data generation has been rather easy for all the platforms. Within 2 months, all the data for these 230 samples were done. The quality of the sample, the QC and the QA is always the bottleneck. And it's always the stuff which people, I mean, don't like to spend too much time on it, but at the end of the day, you pay dearly later. And for research, it doesn't matter. You could always find enough samples. But for clinical, as we've heard today, you can't really, every single sample matters. The analysis of the genotypes allowed us to go from 36 to 95% of the cases that were explainable, either by the MMA or additional genes that were part of the pathways or the regulatory, because it's a metabolic disease, quite quote unquote an easy one. And different roles beside the amino acids have been identified. And this is something that is part of the publication that the guys are working on. However, methods to model these together are still lacking, and they are still living somehow in a silo. And I'm going to use a course that Nicolas Lelovert gave some years ago at the EBI, which was actually, we have models to describe it. And if you look at this from a method point of view, we have a lot of them that could describe the system. We could describe our mutation, we could talk about coverage, etc. It doesn't matter. At the end, we can describe which variant are where with a certain level of certainty. Trying to explain, and Mark this morning showed a very good example of mutual exclusion of mutations. And in this case, the explanation, in this case, the Huxley models for membrane potential, you can explain the models. We are not there yet, not at all in cancer biology and certainly not in other fields. And the one which I tried to allude to today was the prediction. And there we are far, far, far away from getting there. And the prediction is something which I think until we don't take our act, that's more mainly for the more computational biology, the methods developer together and tackle the third pillar, not these first two, because they are and they will be challenged and done in more clinical settings. But the last one is the one which is our next frontier for predicting. And I think that will be our next goal. And with that, I thank you for your attention. I hope I didn't go over time. Thank you.
Well, thank you so much for this talk. No, you are not over time.
Okay,
We
good.
have
4 minutes.
We have 4 minutes left, right? We have 4 minutes left. And let me quickly see whether we have any questions from the panelists, which not quite yet. And the QA session, let me bring that up. So we have no open questions. Yeah, so I was impressed, obviously, by the breadth of your work, and also your work on the kind of building a centralized genomics platform for Switzerland, it seems like, or at least a high throughput facility. I mean, you know, something that always strikes me is like how these prices come down and down to the point where it's commodity. I mean, you must be at the source of knowledge about this. Do you believe that in how many years is your prediction that, you know, it will be almost cheaper to resequence someone than storing his information in an expensive repository? Let's put it this way.
So there's two things. The first one is I think it's going to be always easier to sequence it. But in the same way as the pathology has archive, we're going to have archive for genomics. Now, the question is, how much are we ready to pay for this? My point also is that, and it's true, the genomics is becoming more and more of a commodity. But the point is that it's not the genomics itself or the generation of data that matters, it's how you get to that data. And this one, you could actually try to do that. And I remember a few discussions back when I was still at the SIB about each and every of the hospital claiming they had the best protocol for doing sequencing. And when you really do the round robin and test it, you see that there are gaps and serious gaps, which honestly was tending me not to go to certain type of hospital in Switzerland if I ever get a cancer, for example. So I would say the commodity is there, that's clear. The data generation will be becoming more and more easily, but at the end of the day, the whole interpretative part will be critical. And I have a problem of people giving jobs to each and every of them. I mean, it's easy to put people into a box. And I have a problem with this because one is becoming a servant of the others. My claim has always been for the years, even in the industry when I was on the computation side, and I'm still an immunologist by training, to have an equal partnership, not one that served the others. And this is actually one thing that needs to change and evolve over time here in Switzerland and elsewhere as well.
Thank you. Victor, I think you have time for one quick question. That would be wonderful if that
will be short.
Thank you for this great talk. I think the question is very short. You've raised a key point. You know, every individual institution is too small to really make the digital transformation in Switzerland happen. At the same time, when we look around us in Europe, we actually see big initiatives in Germany. We see big initiatives in the UK, 100 million invested now into digital pathology and transformation. True. So where is the lever that we as scientists need to take to really make politics aware and develop that dynamic in the country so we can really move together with a unified strategy?
Well, I mean, for having done that for the last 10 years, it's going to be a very long answer, but I'm going to be short here because for the next speaker that comes after me. One thing that is clear is our politician, and the COVID actually was kind of a revelator of the lack of understanding for what we are doing. So I think politically, we don't have enough people that are scientifically inclined to understand what we are doing, that they could really see that as an avenue that is critical for the benefit of the Swiss population. They're going to claim it, but most of the time they don't understand it. When Berset, our health minister, was saying that he would not get his genome sequence at one of our meetings down in the west part of Switzerland, it just shocked me, because that means he doesn't understand that there is value in doing this once during his lifetime, and that it will be useful for him later on and for the Swiss population. So I think we need to have the key message towards the politicians, and we need to educate them in the same way as Valerie was saying about we need to train the MDs more towards the genetics and get that inside their DNA. We need to train the politician. That's a whole different ballgame because every 3 years they change. So you need to think one, or we have to get some scientists that go in politics. That's the other solution, which would be the ideas. But that's going to be for a beer session, I would say. Discussion probably at the panel one.
All right.
It's a really tricky one.